So
pour l'instant, je vois pas les gens en arrière, Mathieu. Je sais pas si c'est normal.
we are just waiting a few minutes more to see if there is more people joining.
Yes.
Yeah.
But I think because it's we can open.
Yeah, we already started, Fabien. Yeah.
Yes, I think we can open the session and tell everyone that we are waiting a few minutes.
Yes, yeah. They can see. Yeah, they can see us already.
Okay.
Yeah. So good afternoon, everyone. We're just waiting a few more minutes.
Okay. I think some people will join us later, but we can start. So welcome, everyone. Thank you very much for being with us today. Biotech One, we developed a sustainable process based on supercritical CO2 with application especially on extraction and encapsulation. Today, the session will be led by Elissandro Klein, researcher and specialist in supercritical CO2 at Biotech One. We are also honored to have with us Professor Badens and Adil Mouahid, the inventors of this patented technology. The presentation will last about 20 minutes, followed by the question and answer session. So please feel free also to ask your question on the chat or at the end of the presentation. Alessandro?
Yeah. Thank you. Good afternoon, everyone. Thank you for joining us today. And today we will talk a little bit about our continuous scalable nanoliposome lipid nanoparticle production using supercritical CO2 that we call EncapX. The process and equipment was developed in partnership with the Aix-Marseille University with Professor Elisabeth Badens and Adil Mouahid. So why nanomedicine is so important, why nanoparticles is so important nowadays? From the recent last years, we have seen a huge development in new application, development of new drugs, RNA treatments, vaccine, and premium cosmetics that always face some challenges on the encapsulation part, because the encapsulation is very important to stabilize, to make the compounds bioavailable, and to increase the stability of the compounds. So there is a lot of different applications in nanomedicine, from nanoparticles, from saving life-saving RNA therapeutics to premium dermatology. And nanomedicine has evolved a lot, but manufacturing is not on the same speed of development. So there is still some limitations on the producing a large scale nanoparticles. And the goal is to have high quality RNA therapeutics, lipid-based oncology, and advanced cosmetics. But in the reality, there is some process that are designed for lab, but not really for factory. And the gap here is to find a way to produce encapsulated ingredients in a continuous way, in a reproducible way, and easily scale it up to different scale of production, from lab research to pilot tests, clinical trials, and to industrial production. So the companies always face some limitations on the encapsulating part of the development. There is several different forms, ways to prepare nanoparticles, nanoliposomes, lipid nanoparticles. Some of them work very well on lab scale, but they have limitations for industrial scale, or even some are applied industrial scale, but requires additional steps like the thin layer hydration, for instance. It is currently used on the industrial scale, but it requires additional step after the particle formation to enhance the particle size distribution. So it requires normally sonication, extrusion to achieve smaller and more uniform particles. Um, and it's a labor-intensive and batch-based process. There is also very nice applications of microfluidics that works very well on lab scale, but it always has some problems with clogging issues, considering that during the particle formation, sometimes you use more viscous lipids and so on. And scaling up microfluidics is still a challenge, because to scale up microfluidics, you need to number up the microfluidic devices, and then you also number up the number of microchannels that can have some clogging issues and increase the system complexity. And other option for encapsulation, nanoliposomes production, is the solvent injection, but normally it uses organic solvents and harsh conditions for sensitive APIs. So having that in mind, we developed the EncapX, that is the first continuous supercritical CO2 system to produce in a continuous mode precise, reproducible unimodal particle size distribution of nanoliposomes and lipid nanoparticles. It's very versatile because we can prepare encapsulation from different active compounds, from RNA, mRNA, cRNA, diverse lipids and active compounds, both hydrophilic and lipophilic compounds. And the process is based on a millifluidic technology, which means that we use tubes 1,000 times larger than the tubes used in microfluidics, so we can overcome the clogging issues presented on microfluidics. And the process is based on supercritical CO2, that is a green solvent and does not require any toxic organic solvents for the particle formation. So for those who are not familiar with supercritical CO2, the supercritical state is another state of the matter. So you have the solid, the liquid, the gas, and above certain pressures and temperatures, you can have a supercritical state where the CO2 achieves some characteristics, some properties um, between liquids and a gas. So it have a liquid-like density that enables a high solventing capacity that can modify interfacial properties under pressure. And it also has a low viscosity that enables an efficient fluid mixing and formation of emulsions inside of the device, emulsions of CO2 in water or water in CO2 under the system, inside of the system. So they mechanically basically use CO2 to interact with an aqueous ethanol lipid phase. And reducing the interfacial tension, it promotes the formation of homogeneous pressurized system. And the nanoparticles are formed during the controlled depressurization when the CO2 is released from the mixture. The result is a thermodynamic self-assembled triggered by depressurization, and it doesn't use a share-based size reducing step. So the formation of the particles occurs instantaneously during the CO2 depressurization, and there is a self-assembly formation of the particles. So just to illustrate the process, how it works, we have two streams entering the system. So in the first step, we have a lipids API solution. Normally, we also have a small amount of ethanol to help on the solubilization of the lipids and also lipid soluble compounds. And the second stream is the supercritical CO2. Then we mix these two streams in a millifluidic mixing device that is the patent process. And inside of this millifluidic mixing device, we have the formation of the emulsion that I mentioned before, which reassemble the particles. And then during the depressurization on the third step, we have the release of the CO2 going back to the gas phase, and then the instantaneously formation of the nanoliposomes or lipid nanoparticles in the aqueous phase and the collection in the aqueous phase. And the CO2, as soon as it released, goes back to the gas phase, and we can recycle the CO2 on the system. So with this technology, it's possible to achieve very interesting results with a narrow distribution particle size distribution with a low PDI, very high encapsulation efficiency for both hydrophilic and lipophilic compounds, and is a very robust process, presenting stability across continuous runs and different runs, because we can control precisely the properties of the mixing and of the depressurization step, so we can achieve the same results every time that we run with the same conditions. And the particle size will also depend on the formulation, but the system is very versatile for different formulation, different lipids, different phospholipids and additives. So for example, if you have a good formulation that you tested using microfluidics and you need to scale it up, go for a pilot scale or preparing material for clinical trials or even for industrial production, you can reproduce the same formulation using our process, just optimizing the operational conditions. Here I would just show quickly a case study that was performed in partnership with the Aix-Marseille University, where we encapsulated docetaxel. Docetaxel is a very important anti-cancer drug, but there is some limitations when we use docetaxel because of the low solubility and the sensibility of the molecule. So it's very important to encapsulate the compound to increase the bioavailability and the stability. So we used conditions with 100 bar and 35 degrees Celsius, and we obtained a unimodal particle size distribution of nanoliposomes around between 90 to 120 nanometers, with an encapsulation efficiency higher than 95%, and stability, consistent performance in continuous mode, which allow us to predict an industrial robustness with difficult oncology actives. And we also are performing some validations using different RNA and also some cosmetic ingredients, because we know that for the premium cosmetic market is also very important to protect some sensible compounds like retinoids, antioxidants, and so on. And also for food tech application is interesting to protect some sensitive ingredients. So now we are validating the technology for different applications, and so far we have great results for several different applications. So as I mentioned, one platform that can be used for multiple industries, from pharmaceuticals, RNA therapeutics, oncology drugs, peptides, control release systems, cosmetics, retinoids, antioxidants, botanic extracts, increasing the thermal absorption because of the particle size, and also food tech for aroma encapsulation, natural antimicrobials, nutrient protecting, and so on. And with nanoliposomes, we have some properties that we can protect the ingredients from light exposure, temperature changes, pH change, which is very important for keeping the stability and the integrity of the ingredients at the final product. So a quick overview of the different methods. We have the thin layer hydration, which is currently used in industrial production of different materials, but it is a batch process with low control on the scalability. And sometimes it uses toxic solvents and need to use post-processing technologies like extrusion or sonication to reduce the particle size. Some industries are using microfluidics that can work somehow in the continuous mode, but it's challenging because it requires parallelization, which increases the complexity of the system and increase the clogging issue risk with depending on the formulation. Solvent mixing also can be batch or continuous, but have a moderate scalability, but normally use organic solvents that are a problem depending on the formulation. And EncapX comes with a new approach, a complementary approach for the other technologies, because it's a continuous process that have very high scalability, no microchannels, so there is no clogging issue risks for this process, and it doesn't use any toxic solvents during the formation of the particles, only a small amount of ethanol that we can easily remove at the end of the process. And another aspect that we are validating at the moment is the possibility of use the properties of supercritical CO2 to perform an sterilization in the same process, because supercritical CO2 have the property to sterilize and we are validating also in the next months the application of the integrated sterilization during the particle formation. So we have a patent and validated and ready to sale technology. We can provide the know-how, the equipment, and also assist on the industrialization, on the scaling up, and we can perform proof of concept, feasibility studies, and so on to validate the application of different formulation for different industrial sectors. Biotech One is a deep tech company based in the south of France, where we integrate knowledge in high pressure engineering, mostly supercritical CO2. We use also supercritical CO2 for other applications like extraction, fractionation, sterilization, decellularization, and so on. And we combine also our know-how in process design and lipid science to develop this new technology. And we always have a close collaboration with academic institutions and industrial partners in pharma, cosmetics, and food tech to always be connected with the needs of the different applications of the technology. So with EncapX, we have a continuous controlled scalable process ready for your next formulation. And my question here is what you would like to encapsulate with us. So now I will just open the question and answer. I can also check the chat. So feel free to ask and we'll answer your questions. So let me see. Okay, so I have a question here. Can nanocrystallization of the API occurs while removing CO2? Elizabeth, do you want to answer?
So I have indicated that the composition of the aqueous phase can be adjusted if unfortunately nanocrystallization occurs.
Yeah, okay, yeah. So it can occur nanocrystallization depending on the formulation and on the process parameter. Crystallization, okay. Okay. Yes, anyone have another question? If you have any question, you can switch on your camera and your mic. Or if you feel more comfortable asking on the chat, I can answer on the same way. And of course, if you are interested in our technology, you can click on the book a meeting on the screen, and you can book a meeting with us where we can present more details and more study cases and clarify your questions and also plan some proof of concept or feasibility studies and so on. So feel free to contact us. You can also find more information in our website and in our LinkedIn page. So yeah. There's another question.
Biotech One already carried out clinical batch.
Okay, about clinical batch. At the moment, we didn't perform production for a clinical batch yet, but depending on the needs, we can organize that. We can perform a proof of concept and then plan the production of a batch for clinical trials also. Because with our technology, we are able to prepare a larger amount of the formulation then you are able to prepare with microfluidics, for example. For us, it's easier to prepare a larger volume of the formulation. Okay. Any more questions? And stay tuned, because on the next webinar, we will talk a little bit also about cosmetic ingredient encapsulation. So keep an eye on our LinkedIn page. We will post more details for the next webinar that will perform probably on the next month or in May. And we'll be able to show more results from cosmetic ingredient encapsulation also. If anyone have another question, otherwise I think we can finish. No? Fabien, want to complement anything?
No, don't hesitate if you have some confidential question to book a meeting or send us an email. We'll be very happy to organize a private meeting with the Biotech One team or academic partners. And I would like to especially thank Adil Mouahid and Professor Badens to participate to these webinars.
Yeah.
There's not any more questions.
Yeah, no question anymore. Thank you, everyone.
Thank you.
Thank you. See you on the next webinar.
Goodbye.
Bye.